Yeah.
Is there any part of you, like the kind of light bulb that goes off in my head there is off target gene effects, right? Like, is there any part of you that's a bit worried about, okay, we're going to make it immune to this toxin with this change, but there is a bunch of other stuff that we just don't know what could happen. You know, we know that sort of genes are not as linear as one gene, one function, right? Genes have like interaction effects, gene regulatory networks have all of
Yeah.
these emergent causal properties. Like, is there any part of you that's like, well, we don't 100% know, even if it's unlikely, what trait we could be introducing in the population there after generations of breeding. That artificial selection is going to do something, maybe, but we just don't know what it is. Is there any caution there?
Yeah, yeah, so we think about these things all the time. Like, I think, that's what we're always fixated on as the scientists, is thinking about, are there any unintended consequences that can happen from this? For the toxin one, it's a really simple case that we know there are multiple mammal species that have evolved this same single nucleotide change in South America, but all sorts of different animals have evolved that single nucleotide change that going back for hundreds of thousands of years. And those animals are viable, healthy, population. So it doesn't seem like this one has any deleterious effects. What happens though is every time that gene gets passed on, the genes around it tend to move as a block with it. It doesn't move on its own, like it kind of takes a chunk of DNA with it. So we'd have to be very careful to make sure that when we make that edit, we sequence that animal's genome completely. We can do a lot to make sure that we're not making these kind of mistakes, to make sure there is no off-target effects and that the region of the genome around that looks completely normal. And then you can release that animal. Then the rest of the genes will just kind of mix up within the genome like they would normally. But these are the things that we think about all the time. And then very carefully monitoring that population as it continues to hopefully grow and expand to make sure that, everything stays in balance. And these are things that we can do very well in the environment now, is really track these animals and monitor these animals. We're much better at doing those sorts of things. But I think we've made so many mistakes in the landscape that we have to start thinking about ways that we repair them by
Yeah.
engineering some of these species to face some of the environmental challenges that we've thrown at them. But we have to do it in this very responsible way. So we wouldn't just make the qual, release it out the back door of the lab and be like, off you go, best of luck to you. This would be part of a very carefully controlled release program, monitoring program, really making sure that it's having that positive interaction in the environment. And that would be the same
Yeah.
for every single de-extinction project that we have. It is coupled with a really rigorously designed rewilding project, working with the world's leading ecologists to really look about how you thoughtfully go about putting these animals back into the landscape and monitoring that to make sure there aren't any unintended consequences that come from that.
Yes. I assumed this was something that you have thought about before. I don't expect
Yeah.
any of these. Absolutely. I'm really curious to go into that rewilding part of the process with, say, the Tasmanian tiger. So,
Mm.
am I right to say that in Tasmania, the island itself, it's extinct 80, 90, 100 years roughly or so? But
Yeah.
on mainland Australia, it's much, much earlier. So, like 4,000 years ago or so.
Yeah,
So,
that's about right. Yeah.
so the
So
how would the initial rewilding of Tasmanian tigers happen in Tasmania? There's the first one that is gestated. It
yeah.
has no parents. It has
Yep.
no sort of opportunity for cultural learning. So to
Yeah.
take us from that step to, you know, hordes of Tasmanian tigers hopefully roaming across Australia in a really nice, beautiful way.
Yeah. This is my dream one day, is to be hiking through Tasmania, which is stunning to hike through. And then to see a Tasmanian tiger in the distance is like, I just really hope
I
I
mean, it
get
would
to
be
see that in my
incredible,
lifetime.
right? As a
Yeah.
scientist, to be able to have that much impact, it would be amazing.
Just unbelievable. But I mean, the
Yeah.
thing, to just give a bit of context to the Tasmanian tiger story, the reason this is so important is because it was such an incredibly unique animal. So they look just like canids, like a dog, really, really similar to wolves, but they're a marsupial. They had a pouch. They raised their babies in a pouch, just like any other
Yeah, they don't look
marsupial
like
does.
marsupials. They
Yeah,
don't,
not
well,
at
or at least
all.
it's typical marsupial at all that you would
No.
have in your mind.
Just completely unique in their form. Absolutely unbelievable. But why they were so important for Australia is because they were an apex predator marsupial, and we don't have any others, like no other apex predator mammals in the landscape that naturally existed in Australia. The thylacine was it. And so when we lost that animal from the landscape, it left an enormous hole in that ecosystem. We know from, stories about the wolves at Yellowstone, for example, where they got all of the wolves and eradicated them from Yellowstone National Park, that there was an absolute collapse of the ecosystem in Yellowstone following that. So much so that it led them in the early 90s to reintroduce wolves back into Yellowstone. And then within a decade, it brought that entire landscape back into balance. We've seen the exact same thing happen in Tasmania. So with the removal of the Tasmanian tiger, we've seen very strange diseases like the Tasmanian devil, which is another closely related species, gets this horrific facial tumor disease that would have completely eradicated that population, because you need apex predators there to remove these diseased animals from the population to stop disease spread. And so in the
Mm.
absence of the Tasmanian tiger, you have these diseases just run completely rampant, and it's just completely altering that ecosystem at every single level. So there's a real imperative to want to put a Tasmanian tiger back into Tasmania. And the thing for us, as I mentioned before, we've got a really, really good genome for this species because we've got incredible specimens. So we're able to actually rebuild this animal correctly. There is still the ecosystem that it came from pretty much intact in Tasmania. So there's some really untouched areas of wilderness there that you could immediately put this animal back into, and all its natural food sources still exist. They've all gone out of balance. So there's, more abundant ones and less abundant ones, but everything it needs to survive and thrive again is still there and intact in Tasmania. So it's a really, really good project for why we would want to do a de-extinction. And of course, it was a completely human-driven extinction. We hunted these animals brutally to extinction very quickly, because when colonial people settled in Tasmania, they thought it was very similar to the European wolf. It was going to kill their livestock. So they're like, we want to eradicate these species, immediately. So they got rid of them very quickly. Now, when it comes to rewilding and creating those first animals, you're right. The first ones would either be made in a surrogate animal, which is probably going to be a very small marsupial called a fat-tailed dunnart, which obviously would not be suitable for raising a full adult thylacine because it would probably eat it pretty quickly.
It being like
Or,
one hundredth the size of it, right? I
yeah,
saw a
yeah.
picture. When I was researching this, I was like, this is the closest living relative. I looked at it, I was like, that can't be right. I must have misspelled. This thing is like miniature. It's a tiny,
Yeah,
tiny little mouse, field mouse sized thing. And it's
yeah,
gonna birth an animal that goes, what is it, maybe like 20 or 30 or 40 times its body size in
yeah,
mass? Like it's
absolutely right.
much like that's a
Yeah,
bizarre thing, isn't
yeah.
it? That's very just a
And
strange
this is
thing in of itself.
one of the magical things about marsupials, right, is they have tiny, tiny babies. So
Yeah.
the marsupial baby at birth is about the size of a grain of rice. So that means even our little field mouse-sized dunnart can actually give birth to a baby thylacine. It's one of the incredible things about them. And it would
It's
outgrow
wild.
mum's pouch within a few weeks, but it could certainly survive in that mum's pouch for the first few weeks. And then you could remove that baby. They're about the size of a jelly bean when we'd have to take it out. And then, or, a baked bean or something around that size. And then you can bottle feed them to actually get them up to that independent level. Now, one of the good things
That's
about
crazy.
marsupials, from our perspective at least, is that they do have relatively small brains. It's a bit smaller than a placental mammal, which is all the other mammals that you can think of. So lions, tigers, cats, bears, all of those things. They're called placental mammals, different from marsupials. Their brains are much bigger. So that's why we have a lot of domesticated placental mammals, because they are quite intelligent and, you can train them to do things. We don't have any domesticated marsupials because they don't really have a good capacity for that. But what it means is that their brain is much more hardwired with a lot of these, really pre-programmed behaviors. And
Right.
some of the most hardwired behaviors, even in our brains, are your feeding behavior. So what it is that you want to eat, and also your mating behavior. There are two things that are absolutely hardwired in any animal's brain. And so, we know this from rearing orphan cubs, that people will do regularly, or any orphaned animal, that you can put those animals back into the wild and they do innately know the core behaviors for their survival, which is a really good thing. But marsupials have much more pre-programmed and much less capacity for learning. So we do believe that those first few animals will be able to do it. A great example of this is even with domesticated cats that, we've raised in our houses for hundreds of generations now, feeding them tin cat food. But if you wiggle a feather or something in front of them, they immediately know that they're pounce predators. They'll leap out of nowhere and jump onto it and try and rip the thing to pieces. And that's because those behaviors of you are a pounce predator is absolutely programmed in their brain, even after hundreds of generations of domestication. So you can imagine an animal that's been cloned from a wild animal is going to immediately have those pre-programmed behaviors. But you can also train them. So in the cases where people have raised orphan cubs and they're not quite up to the task of, understanding how to kill a prey or, eat a particular thing in the landscape, you can actually train them to do that as well if it's necessary. So the first few thylacines that we would make would absolutely be raised in captivity in a very large outdoor area, similar to what the diables have, I would imagine. So very large, open roaming area, but very well fenced so that we know exactly what's inside the fence as opposed to outside of the fence. And you would study very carefully those animals' interaction with the other species within that enclosed system. We would study their behavior and make sure they are feeding okay, they are breeding, and hopefully doing all of the things that we would hope that a Tasmanian tiger would do. And then from there, you would do progressively bigger areas that you would fence off. And again, very closely monitor that these animals are having a really positive impact in that ecosystem. There are things that have changed in Tasmania since we lost the Tasmanian tiger. One of them is there's a lot more cats now, unfortunately. So these are domesticated cats that have gone feral, so they've gone back into the wild, and they're wreaking havoc with our wildlife. And so we want to make sure that that additional pressure of cats in the environment, as well as Tasmanian tigers, isn't going to push some of those species that are on the brink of extinction even further down that road. So these are things that you
Right.
have to monitor very, very carefully. One of the great things we do know is that cats hate other large predators in the landscape. So if there's a fox or a dingo, for example, in that same area, cats will leave that area. So one of the things that we're hoping is when we put thylacines back into some of these densely forested areas, so that's enough to put the cats off and make them move out of some of those areas. And of course, the Tasmanian tiger is so much better in balance with all of our native wildlife. It can't climb trees, it can't go into small holes to kill, smaller marsupials. So it's in much better balance with our landscape and hopefully pushes those cats back out into more of those urban areas where they can do less damage. So I guess we'll wait and see, but certainly dingoes and foxes have that effect. So we're hoping that a Tasmanian tiger would do the same thing.
It's so fascinating. From a complex dynamical systems perspective, it just seems like all of the interacting parts in an ecosystem are so bloody complicated. And
Yeah,
there's so many more interactions going on than we've possibly even mapped to this day. Like we don't even know how all of these pieces are fitting together. But I guess what you're saying is that we can monitor the situation so closely on
yeah,
so many levels constantly and be looking for all of these different emergent interaction things that are popping up like, oh, that's strange, that population of that herb is going further, or the
yeah,
disease is spreading. So what you're saying is like it's very complex. Maybe we can't map every interaction or everything that's going to happen, but what we can do is observe it so closely that when something is happening, we can act on it quite quickly and make changes required. Is
yeah.
that sort of roughly the idea?
Yeah, that's exactly it. And we know these landscapes are better with those species in them than they
Yeah.
are trying to survive without them. And that the longer the Tasmanian tiger is missing from Tasmania, the more that ecosystem is going to fall apart. So we know the quicker we can get those animals back, the better off we are going to be at really bringing that ecosystem back into balance. But you're right about that complexity. I mean, there's some fantastic documentaries you can watch about the removal of the wolves from Yellowstone, and one of the profound things that, they really documented from that is that the river changed its course through Yellowstone Valley as a result of the loss of the wolves. And that was
Right.
because once the
Exactly.
wolves were gone, then the deer population exploded. The deer are eating all the new shoots and growth off of all of the new bushes. So without that, there was no flowers. So the bees left, then the bears left. Then the deer had nothing left to eat. So they moved down to the edges of the river. They started eroding the banks of the river. The beavers left because the water became too dirty and it was not healthy for the beavers to be there anymore. Once the beavers left, the river just changed its course through the valley. It just wasn't following its same path it had followed for thousands of years documented before that. And then within 10 years of the wolves being put back in that landscape, everything changed. The birds came back, the bees came back, the bears are back. Even the river regained its original course through the
It's
valley
wild,
because
isn't
the
it?
beavers
Literally
came back in
wild.
again. But, for us as scientists, I don't think we'd ever really understood that level of complexity, that it's changing the physical landscape. Like, you think
Yes.
you remove the wolves and, like, sure, the deer populations are going to explode. Like, we would go like, yeah, that makes a lot of sense. But then really understanding the complexity of the knock-on effects of that for every part of that landscape was incredibly profound to understand from that example. And because it was a national park, it was exquisitely well documented by all the park rangers who were out there all the time. So it's probably the best example of just really getting that understanding of how the removal of one species can make an entire ecosystem fall apart, and why it's so important that we preserve every component of biodiversity to make sure that we're not in these situations where we have to re-engineer that ecosystem back again.
Yes. Such a good example. It highlights exactly what I was talking about.
Mm,
Just this complete knock-on effect that you would never possibly, and you
yeah,
couldn't even calculate ahead of time.
no, it's
If
crazy.
if we're if we're talking about the genetic diversity of the new Tasmanian tigers, how is that going to work? Because obviously you produce one and it has its own genome. Then the
Yeah,
second one that you produce, or the third or the fourth, you're trying to vary those genomes as much as possible, right? Because they have to eventually mate with each other and
yeah.
create the whole population. So, like, how do you think about getting the right level of genetic diversity in those populations where too much genetic diversity and it stops kind of looking like a Tasmanian tiger anymore, too little and it's vulnerable to environmental change? Like, what? How does that work? Because I'm trying to think.
Yeah,
Like, do you need
again,
like,
this is something I think about all the time, right? So,
yeah,
so the way that we're approaching it again is because we can do this in a stepwise fashion. So, initially, we're sequencing the genomes of about 50 Tasmanian tiger genomes. Um, Most of them were collected right around the time that the animal went extinct, so in the early 1930s, and the animal went extinct in 1936. So it was right around that when the population was just about to go extinct. And so we have a really good understanding now of what the sort of contemporary diversity looked like, if you know what I mean. So what that diversity of the population looked like before they were hunted to complete extinction. And then we have our older specimens. So some are 100 years before that, and some of them are even earlier than that. And then we've got some animals that were collected from fossils, or not fossils, but from specimens that were found in caves on the mainland. So they're the order of maybe 6 to 10,000 years old. So we've got all of those animals now that we can sequence genomes from. So that gives us an incredibly rich pool of genetic diversity. So initially, what we would do is we're going to engineer back in the more contemporary diversity. So what the diversity looked like in that population right before they went extinct. And they were being hunted to extinction, not naturally going extinct. So it wasn't like, they'd lost a lot of diversity or anything. They were just being eradicated because we were going out and hunting them. So we'll re-engineer as close as we can to the health of that population that existed right before they went extinct. And that would give us the most recent snapshot of diversity. And then again, like we were talking before, monitor those animals and see how they're going in the landscape. Are they able to adapt to different environments? Are they moving into different areas? Are they breeding really effectively? And then we can sequentially add more genetic diversity. This bit's relatively easy compared to re-engineering, the missing bits of the genomes, for example. That's a really big job. Putting this diversity back in again is much, much more simple. And so we can go back and keep on adding more diversity until we get that really healthy population. But you're right, you don't want to go too far either. So if we don't have to go back to 5,000-year-old, 10,000-year-old diversity from mainland Australia, great, we would never have to put that in. If they're not doing so well, we might think about putting some of that diversity in. Or if we decide it might be really good to try and rewild some of these animals back onto mainland Australia, then we can add some of that diversity that came from the mainland back into that population so that they can hopefully exploit a wider range of habitats on mainland as well. And there's lots of good reasons why we might want to do that. One of them being that we have a massive fox problem that was introduced from Europe. They're not native to Australia. They're very problematic. And so it would be great if, again, that thylacines could help push foxes and domestic cats out of different areas of the forest. Because, again, foxes don't really like there being other big predators around. They're quite adverse to that. So they could be a very useful tool for helping, help combat some of these pest species problems, even on the mainland.
yes. I find all of this so interesting, and you're really good at communicating all this. I mean, this has been so much fun. I've learned a lot. Just you said it's very simple to increase genetic diversity there. Explain how that process works. you print one with a high level of genetic diversity and kind of send it out into the population, and that kind of spreads through it over time? Or like, how does that process
Yeah.
work?
Yeah, so that is how it works. But actually, you could engineer, 20 animals' genetic diversity into just one animal. So you can put all of those differences that you see between of those and make one animal that's got a lot of differences. But then
Right.
when the animal breeds, we mix up our chromosomes. That's why we're all a blend of what our parents look like, is because their chromosomes kind of mix up before they're segregated down to us. And so if you engineer a lot of diversity into one or two animals, it will then get separated out and segregate out among its offspring. So there are ways of doing this really efficiently, that you can incorporate a lot of genetic diversity back into a population relatively easily. And that genetic diversity, there's not a huge amount of differences between any two animals' genomes that give them that genetic diversity. So it's not a lot of engineering to put those differences into that genome to give those animals that diversity. And like you said before, this is really what is critical for them, surviving diseases, but also environmental change, things like global warming and stuff. The more diverse an animal's genome is, the better its ability to adapt to rapid environmental change. So it is something that we know species today need as much genetic diversity more than ever. So that's why I was saying before that really important use of de-extinction technology to put that diversity back in. It's because we need it now more than ever, because we're
Yes.
changing the planet at a rate that's never been seen before. And our poor wildlife is really struggling to evolve quickly enough to keep up. And it desperately needs that genetic diversity just to give it a greater selection of things it can choose from to become the most optimum form for how quickly we're changing the planet.
The Giant Shoulder mission is to explore radical ideas in biology, neuroscience, and consciousness, and elevate those stories to the highest possible level while keeping them accessible to everyone. If this interests you and you want to support independent science, then please consider subscribing to the Clips channel. Check out our 26 Neuroscience book. You can download it for free